So we talked in general terms about the cones and the rods, but we can go a little bit deeper and see where exactly are they found. For example, we said the rods and the cones are found in the retina, especially in the photoreceptor layer of the retina. So let's look at the cones again. Yes, they are found along the retina, but they are concentrated around the fovea of the retina. Okay, they're very good in light, in well-lit environments, or when you have sufficient light, the cones are good to use. So they operate in daylight conditions, high light intensity. They're also very good at color differentiation. That means that they are the ones that help in us perceiving colors properly. If you're looking at a black or red or yellow, the codes are good at that. And then they're also very good at high visual acuity. And that means that you can differentiate between two close points because of the cones. Okay, and so the reasons, of course, we shall look at color vision and see what theory do we have that explains the color vision. And then we also, we have already explained this in terms of the fact that the cones are connected to, each one is connected to its own bipolar neuron. And that means that the details that you get about an image are actually, the details are improved. Okay. But also they are tightly packed, and this individual connection is what I've just explained. Connection of each cone connects to its own bipolar neuron, and this is what we are explaining here. Okay, but also the tight packing is very important because you will have several of them, and then that actually increases the clarity and issues also to do with visual color vision. The rods, for them, they are located in the peripheral of the retina. That is one, not centrally. Okay, they are around the edges or the peripheral. Very good when you have low light, and they contain rhodopsin instead of iodopsin that is contained in the cones. The unique feature with rhodopsin, which you really know that it is a photopigment, is that yes, this rhodopsin is actually broken down, and then later on it recombines to regenerate. Okay, but that regeneration is very quick. So that is also another difference that you need to take note of. Regeneration of rhodopsin is very fast compared to that of iodopsin. And the other thing that the rods have what you call retinal convergence, where you have one rod, several rods connecting. You said we have several rods connecting to one bipolar neuron, and this is what contributes to high sensitivity, but also the disadvantage is that it lowers visual acuity. Okay, so if we were to repeat everything that we have talked about, you have cones more around the fovea, and then the rods, we don't have a specified point where you have them highly concentrated. And then, of course, the nails, rods, and cones, and the names arise from the shape that they have. The cones are cone-shaped, the rods are rod-shaped, and the cones are larger compared to the rods. And in terms of photosensitive pigment, iodopsin is what we have in cones, and this iodopsin is in 3 forms, and then rhodopsin is only in 1 form in the rods. And then you have the cones being sensitive in high intensity light, that is during day, and then the rods are very good in low intensity environments, especially at night. And that means, and then we move to the cones are very good at color perception, and then the rods do not distinguish between the different colors that we have. Rods have what you call synaptic convergence. Cones don't have. You have one cone per bipolar neuron. That means, and that is actually what results into high visual acuity in the cones compared to the rods. In terms of what they are similar, both of them, both rods and cones, are photosensitive cells. They are found within the retina. Their structure is that you have all of them actually have a constriction. They have cilia, and then the inner segment is also similar. They have the same organelles, mitochondrial nucleus and other organelles. They are also connected to bipolar neuron. The difference comes in how many bipolar neurons are they connected to. And then they have photosensitive pigments that are bleached by light energy. And then both of them are located in the retina and have photosensitive pigments. So that marks the end of this presentation, where we have looked at the rods and the cones, and the fact that you have the rods being very good at night, and then the cones being very good during day when they have light intensity. So we move to light perception and how that happens. Thank you. You see this, this here. You have 3 rods that connect to one bipolar neuron. That is what you call a synaptic convergence. And that makes the rods actually very sensitive. But also it has a disadvantage of making the rods not to be extremely very good at visual acuity. So that's the challenge there. So that means that the cones, since each cone is connected to one bipolar neuron, they are very good at visual acuity. The visual acuity is the fact that you have the ability of the eye to be able to distinguish between 2 close points. That is visual acuity there. Okay, so that means that the cones are good at visual acuity, whereas the rods are bad and not very good because of that synaptic convergence. But the synaptic convergence also gives the rods an advantage over the cones, because they actually make the rods very sensitive to light. That means that they actually can work in dim light. That's what we emphasize there. And we earlier on talked about the horizontal and amacrine cells being involved in what we call lateral inhibition and visual processing. So let's basically understand what that means. So when you have light striking the photoreceptor, you will have different reactions that will happen that will result into an electrical signal being transmitted to the bipolar cell, so the bipolar neuron. And so that means that excitement will progress until it reaches the brain. But one of the things that happens is that when you excite, let's assume these are photoreceptors cells, if you excite this photoreceptor, the neighboring photoreceptors are inhibited so that they cannot fire electrical or electrical signals. Okay, so that means that they are inhibited from working, and that improves the image that will actually be formed by this excited photoreceptor as the information reaches the brain. So it makes the excited photoreceptor to actually enhance what you see. The sense that you have, you can easily distinguish between or contrast contrast between light and dark areas. Okay, so that means that if all these are allowed to work simultaneously, then you may not have a very good, well contrasted image. But if you inhibit this and focus on this one, it gives you a better, strong response, then that is better. And that is what we call lateral inhibition. So the lateral inhibition is because the horizontal cells inhibit, they're the ones to inhibit. Say, please don't let this one be the one to move. Okay, so the electrical signal goes and it improves the contrast of the image.